Scientific methods. Table of contents. Observations and experiments. Measures of data. Graphs. Normal or parametric distribution. Chi-squared test. And designing a simple experiment. Observations and experiments. Scientific progress can be made from observations and experiments. Charles Darwin and Alfred Wallace made careful observations about biodiversity and developed the theory of evolution. Observations, or observational studies, take measurements or surveys of a sample without trying to affect them. Gregor Mendel carried out experiments on garden peas and developed the rules of heredity. In experiments, scientists carry out tests, trials, or procedures by manipulating variables to see the effect of each change. When you perform a biodiversity survey on a plot of land or measure a rate of diffusion, you are carrying out an observation. When you study enzyme activity or measure rates of transpiration, you are carrying out a controlled experiment. In either approach, carefully analyzing data and minimizing error and bias are critical. Experimental design. Typically, an experiment tests how changing the value of one variable affects the value of another variable. The variable the experimenter changes is called the independent variable. The variable which changes as a result is called the dependent variable. The designation as to which variable is which is not due to nature, it is due to the design of the experiment. So, any experimental design has to make it clear which variable is being adjusted by the experimenter and which one is being monitored. An experimental setup may have a number of potential variables. It's important to change the value of just one variable at a time, the independent variable, and not the others. Otherwise, it would not be clear which change resulted in an observed effect. The variables that are held constant by the experimenter are called control variables. Questions, hypotheses, and theories. Observations, research, discussions, etc. can lead to new scientific questions, hypotheses, and theories. A question provides a clear path for research. It arises from curiosity about a natural phenomenon. A hypothesis is a statement that proposes a testable answer to the question. A theory is an explanation of a phenomenon that has been repeatedly tested using scientific methods. Background research. Background information is required to develop a rational hypothesis and experimental design. Scientists rely on information gleaned from those who have completed experiments on similar topics in the past. This helps science progress. New knowledge is built from prior knowledge. Everything doesn't have to be rediscovered by each new generation of scientists. Minimizing experimental error. Sampling error describes the case when a sample is not an accurate representation of the total population. The sample may be too small or not random. For this reason, in a biochemistry experiment, scientists run multiple samples for each condition and repeat experiments. Measurement error describes the case when measurements lack precision or accuracy. Qualitative data is prone to measurement error since it is subjective. Therefore, quantitative or numerical data derived from scientific instruments are preferable. Minimizing bias in research. One source of error is bias on the part of the researcher. Bias may arise when a researcher is influenced by their desired or expected outcome. This bias can impact experimental design or data analysis. Careful researchers always search for possible alternative explanations to their findings. Qualitative data requiring human judgment is especially prone to bias, so should be minimized. Data should be analyzed in a blind fashion by setting it up so that the experimenter is not aware of which conditions apply to the data being analyzed. Control samples. Controls are reference samples that ensure that an experimental system is working reliably. An experiment may have many controls and may be referred to in many different ways. Control samples, control conditions, control treatments, baselines, standard treatments. A positive control sample has all the necessary variables and all the necessary conditions required for a measurable change to occur. It's a sample that should work well and give a strong result. If it fails to produce the result, the system is not working. A negative control sample lacks a crucial element required for a measurable change to occur, and there should be no measurable result. It is a sample that should not work. Any measurable change in a negative control would indicate that there are unintended variables impacting the experiment, the results are erroneous, and the design should be reviewed. Standard curve. A standard curve documents a series of reliable experimental results in the form of a graph to which comparisons can be made. It is also used as a reference tool to quantify results.